adventures unfurl beneath sterile light, where the day stretches into a slow and soothing study of tiny lives unfolding at their own unhurried pace. If you find comfort in this kind of gentle journey, you can softly tap like and subscribe. And if you want a longer drift into calm learning, there's a playlist linked in the description that will keep you company while you rest. What city country are you watching from right now? You find yourself by a clean bench, the surface gleaming softly, a faint reflection of the overhead light stretched into a quiet glow. The room is arranged with comforting precision. Rows of labelled bottles, stacks of clear petri dishes, everything purposeful, everything serene. A laminar flow hood whispers with filtered air, creating a simple protective breeze, the sort of tender background sound that loosens tension in the shoulders and slows the breath without you noticing. Your hands move with unrushed care, gloved in powder-free nitrile that hugs the skin in a smooth, matte touch. The slightest scent of isopropyl alcohol, cool and clean, drifts lightly in the air. It's not sharp, it's refined and soft, a signal to the senses that everything is in order. A petri dish rests beneath the gentle light. Its base is filled with firm, pale agar, nutrient-rich and calm, a glassy meadow for quiet colonies that will appear in their own time. You lift it with a steady hand. The lid slides slightly, a whisper of plastic touching plastic. The dish is labelled with neat handwriting. Date, initials, a short code for the medium. The letters are plain and comforting. It's like a sleeping face you check on as you pass the crib, making sure everything is peaceful. The world beyond these walls may move quickly, but here, time expands and becomes soft. Every moment lengthens and settles, and this slow stretch of minutes feels kind, as if it understands your need to rest. A loop awaits near the burner or the quiet electric sterilizer, its slender wire curving like a drawn-out breath. You hold it steady and warm it, not in a blaze but in a disciplined, patient way that respects the delicate state of your soon-to-be companions. When it cools, you dip it into a small tube where the culture rests like sleeping tea, cloudy only in a gentle way. You touch it to the surface of the agar. The first contact is subtle, like a fingertip reading the grain of silk. Then you draw a line that is not a line, but a soft trace, a glide across the surface, easing the cells out so they can find space to grow alone. The motion is familiar and slow, a quiet choreography. Streak, turn, streak, turn, streak again, your movements diminishing until they become mere whispers. You imagine the cells settling down, separated by a careful hand, each with a tiny patch of room, each with a chance to begin. As you place the lid back on the dish, a dew of condensation beads near the edge, catching the light like faint stars in a pale sky. You don't rush, you tilt just slightly, letting a tiny shift gather those beads away from the center. Your breath remains easy. The dish goes into a waiting incubator with a soft click, joining others in neat stacks, each one a quiet story that will reveal itself in slow awake. The incubator hums contentedly, a small warm space rest your hand on the handle, warm, smooth, and feel the idea of growth humming through your fingertips, so gentle you almost think you dreamed it. The hours unfold like a long, peaceful corridor. You write in a notebook with lines that comfort the eye. The page accepts the motion of the pen with barely any friction. Date, time, sample, medium, temperature, expected outcome, small observations. The letters trickle out of your hand like a calm stream. You describe what is and calmly what might be. You do not race to conclusions. Here, in the patient hours of a microbiologist, the desire to hurry melts away. The gentle rhythm continues. Imagine yourself moving to a clear bottle of media, the liquid golden or pale straw, balanced at a quiet simmer in a water bath or resting at room temperature, ready to be poured into waiting dishes. The agar had melted earlier in another patient hour, and then cooled just enough to flow like honey in winter. You lift it with both hands, slow and secure, and pour. The stream is smooth and unbroken. It slides across the surface, rising to a thin, even layer that stops just before the rim. Dish after dish receives its calm portion. The room smells mildly of agar, like steamy rice, but more delicate, a scent that evaporates quickly as the plates set, turning from glossy to faintly matte, like a still pond under overcast skies. When the last plates have settled, you lay them with their lids half ajar for a minute, a quiet ritual that lets any lingering warmth drift away. Then you lid them and wrap them in small stacks or slip them into sleeves, labeling every group with unhurried neatness. The gentle rhythm continues. You imagine yourself returning to the incubator, listening to the sound of the fan that moves air in the background, the soft hum harmonizing with your breathing. There is nothing to fix right now, nothing to solve at once. The work is a steady path, and the path is comfortable beneath your feet. A timer goes off somewhere not as a shrill alarm, but as a discreet tap on the shoulder, just a small chime that fades into silence. It's time for a check, not a conclusion. You pull open the incubator, the door giving a quiet sigh, and you lift the first plate. On the agar surface, small circles have appeared, barely larger than tiny beads. Some are pearly, glinting under the sterile light. Others are matte, like small clouds. A few colonies show a faint halo of a different texture around them, almost like a blurred glow. Each shape has its own mood, convex and smooth, or flat with a gentle reach, or wrinkled like a soft button. You lean in without hurry, taking in the subtle gradients of cream, pale yellow, butter white, a breath of beige. It is soothing in its variety. Each colony seems like a whisper from hours you already lived, and hours still to come. Time made visible in quiet, round notes. You record what you see, not because it is urgent, but because it is faithful. In the notebook, you describe the edges as entire or slightly undulate, the surfaces as glistening or dry. Words like convex and mucoid sound less like jargon and more like the soft textures of rain and bread dough. You note that some colonies are closer to each other, neighbours on the same street, and others stand apart in open spaces. You look at the base of the plate and see the faint lines of your streaking, now a map of movement from earlier in the day, still present, still telling a story. The gentle rhythm continues. Imagine a broth tube that was clear a few hours ago, now holding a delicate cloudiness, a veil suspended in the liquid. When you tilt it, tiny flecks move like pale snow in slow motion. The bottle returns to upright, and the snow settles back into a steady suspension. You might measure the density gently, placing a small cuvette into a spectrophotometer with a satisfying click. The machine offers numbers, but the numbers feel like soft pebbles rather than hard stones, solid, real, but not demanding. They quietly reflect what is unfolding. You jot them down with the same calm hand, ink meeting paper in quiet strokes. At the bench, a pipette rests like a slim metronome for patient time. Your thumb presses the plunger, feeling the smooth two-step resistance. A familiar gentle press. You draw up a small volume of clear liquid, 10 microliters, or maybe 100, crystal beads in a transparent tip that catch the light as you move. Each droplet is a world of potential. You release it onto a plate or into a well, the way a quiet rain touches a garden. The pipette tip taps the plastic with a soft sound, almost musical, then lifts back and is ejected into a waste container with a mild click. The action repeats again and again, an undramatic ritual that brings peace with its predictability. You imagine yourself labeling a tube with a fine marker, writing so carefully that each letter is almost a piece of art. The tube nestles into a rack, snug and stable. Around you, the subtle green or blue glow of the equipment's indicators reminds you that everything is alive in the gentlest way. The incubator warming softly, the shaker turning with a quiet wash as flasks ride their tiny waves, the laminar hood maintaining its calm breeze. Your breathing falls into line with these rhythms, the inhale paired with the glide of the shaker, the exhale paired with the steady hum of a fan. The gentle rhythm continues. You glance at the clock, and the passage of time in this place is like watching a shadow move across a wall. It's not a rush, it's a glide. Minutes become hours in a soft way, and the day becomes evening almost without an edge. Under the sterile light, your mind feels both occupied and unburdened. The details are just the right size, small enough to be simple, precise enough to be satisfying, and never too loud. The room keeps you company without asking for attention. Imagine a plate that yields just one colony of interest, a single small circle of life that calls your hand to it without insisting. You lift the lid, slide the loop again, and touch the colony like a borrowed ember. It transfers to a fresh plate, where you draw your careful pattern again, giving it space to rest and to bloom. You do not hurry. You honor the interval. With each pass you move further into clarity, away from a crowd and toward a soft kind of purity, not sterility as a harsh emptiness, but a gentle clearing where one presence can be understood by another. As the hours letters unfold, you pause for a sip of water in a quiet corner of the room, the glass cool in your hand. You listen to the steady hush of the hood, the distant tick of a timer, the almost inaudible drifting sound of airflow. You think of how these tiny living travellers move through phases, much like a day does. A beginning where they settle in and learn the environment, a growth where energy is abundant and the edges expand, then a pause when resources grow thin and everything becomes still. The storyline is not tragic or triumphant. It is cyclical, restful in its predictability, like watching a plant unfurl a leaf in spring and fold its petals at dusk. You briefly imagine a plate of colourful cultures made safely from harmless strains that play at pigment, soft pinks, gentle golds, a breath of deep green under natural light. They remind you that even at the smallest scale, variety is part of a quiet beauty. No one colony is loud. Together, they compose a peaceful choir, each voice calm, each texture a muted note. The gentle rhythm continues. At your side is a small flame or an electric glow, a symbol of cleanliness rather than spectacle. You use it not as drama, but as assurance. You pass a metal loop through it, let it pause to cool, and return to the work. The movement is deliberate, the way you might fold a blanket to smooth creases. The flame vanishes from your thoughts almost immediately. What remains is the soothing feel of doing something in a careful ritual way, so that the next action is as quiet and safe as the last. Over time, you find yourself thinking about the rest of the world in gentle parallels. The agar plates are like gardens, shallow and peaceful. The colonies are like small shrubs, round and tidy. Broth cultures are like mist in a glass jar. The incubator is a sunrise that never quite turns to noon, just a warm, steady morning. The shaker is a calm tide, lifting and settling, lifting and settling. The notebook is a chronicle of kindness to detail. These metaphors soften the edges of science and make it feel like a lived-in room, where every object has a place and every process has a pace that respects the breath. Imagine now that you are pouring plates again, but this time you are thinking about what is in them. The medium is a simple recipe: water, nutrients, a source of carbon, salts, and gelling agent. Each ingredient is modest, yet together they make a calm home. You might think of it like a classic broth, but transparent and light, a gentle invitation rather than a command. pH is adjusted with patience, a drop at a time, until the solution is balanced in a way that feels quietly correct. The gentle rhythm continues. Later, in a different corner, you prepare slides for a simple stain, not a harsh display, but a tranquil reveal. A drop of liquid rests on glass, a thin moon shimmering in a still pond. You let it dry and fix it gently. The dye touches it with a soft wash of colour, then clears away, leaving details behind like footprints in fine sand. Under a microscope, these details come into view with the turn of an unfussy knob. At first, the image is a fog, then it becomes crisp, small shapes, ovals and rods, floating like discrete strokes of ink on parchment. They are not threatening, they simply are. are short and neat, that the clusters are even, that the surface of the colony showed a hint of shine under the light. You include the temperature, the time, the medium, the method. These are all parts of a gentle equation that does not demand a solution tonight. It is comfort to know that knowledge grows in increments, in patient layers, the way a pearl forms in calm water. Imagine stepping into a walk-in incubator, temperate, softly warm. Storage racks stretch in quiet parallel lines, each one holding trays that look like simple landscapes. The floor is clean underfoot, and even the sound of your steps is muted, as though the air itself wants to be still. You check the log, numbers, times, conditions. The data is steady. You close the door softly, and the warm hush resumes, like a room inhaling in slow motion. Back at the bench, you set up a PCR machine, its lid gently heavy, its interface smooth beneath your fingers. You think about cycles, denaturation, annealing, extension, the gentle repeat of temperatures that coax a small region of information to copy itself. You measure tiny volumes with the same calm thumb on the pipette plunger, mixing ingredients that look like water but hold subtle purpose. The tube's cap closes with a soft click. You place it in the machine. The program begins, and the small screen glows. The hours will pass whether you watch them or not. The gentle rhythm continues. In the quiet, you decide to tidy the bench. Wiping the surface with a clean cloth is a simple joy, a slow, even sweep that leaves a faint, pleasant scent. You align the pipettes, check the stocks, restock the tips. The waste container is emptied with a careful hand. The trash bag rustles softly. The bins are lined again with crisp, light plastic. The room feels refreshed. You sit for a moment and simply breathe, listening to the hum that never seems to tire. Your eyes soften, not from fatigue, but from the comfort of repetition. The door opens and closes with hardly a sound. You picture a colleague moving just as gently somewhere nearby, their footsteps a companionable cadence. A brief exchange of nods says enough. The work is easy to share in silence. The culture of quiet extends to the people as well as the microbes. It is a soft community, supported by the stable certainty that time and care make results, just as surely as warmth and nutrients make colonies. The gentle rhythm continues. Imagine now that a new batch of plates is coming out of the autoclave cycle, warm, safe, ready. You take a moment to appreciate the reliable transformation inside that closed chamber, heat and pressure cradling glassware and media, leaving them clean and simple when they return to you. The door opens slowly to let the warmth roll out like a heavy sigh, and you keep your hands a small, respectful distance from the gentle fog, letting it fade before you step closer. The metal tray glides onto the counter with a heavy but muted sound. You lift each item with a care so instinctive it requires no thought. You find yourself curating a small collection of examples for students who will come tomorrow. Not a lecture, not a performance, just a quiet display. Plates showing concentric rings of growth that arose from a swab of soil. A dish with a feathery edge reminding you of frost. A broth that turned a subtle shade in response to a tiny change in pH. You arrange them with the care of setting a table, each item given space and a label. You imagine a gentle conversation where someone asks with simple curiosity, and you reply in kind. The subject is the sound of life, not its noise. Later, the microscope calls you again, not with urgency, but with a soft tug. You place a slide beneath the lens. Focus drifts in like a tide, bringing tiny forms into view, then pulling away and returning with better clarity. You linger. You think, with a sense of calm, how these shapes have been present on Earth for so long, not as villains or heroes, but as threads of a vast fabric. Some threads weave into our own lives closely, becoming companions in our digestion, our soil, our kitchens, where bread is encouraged to rise and milk is encouraged to transform. You picture a jar of yogurt thickening overnight, a loaf blooming in a bowl on a counter, sauerkraut brightening in a crock with a faint friendly fizz. The sense of connection here is not heavy. It is nourishing and light. You check the incubator again. The colonies have grown slightly since the last glance, enough that the edges soften into confidence. You consider the next step, maybe a small picking of a single colony into a tube to start a fresh culture. The motion is so familiar that your hands seem to perform it by memory. The colony breaks away from the agar with almost no resistance, like a dewdrop lifting onto a blade. It disperses in the clear broth with a few swirls, and the tube rests at an angle in a small rack, its label solid and precise. The gentle rhythm continues. Imagine now a marine sample, a small bottle with seawater that looks as unremarkable as a glass of rain. You bring it under the hood. The smell is faintly mineral, almost not a smell at all, more like a suggestion of salt carried in the gentlest breeze. You filter it through a membrane, feeling the slight draw of vacuum, a tug similar to the wrong side of a yawn. The filter turns speckled with the tiniest dots. You lay it onto agar designed to imitate the ocean's embrace and tuck it into the incubator. Time will draw small constellations there, as quiet as plankton drifting under moonlight. The day continues like a steady line that never wavers. You sit for a moment and imagine the immense patience of the processes around you. The microbes do not hurry, and their calm is contagious. They settle, they divide, they pause, they listen to the environment's message of warmth and space and simple food, and they respond in kind. You think of their growth curve as a simple and soothing melody, a slow beginning, a rising swell, a plateau of contentment. It's a shape the mind recognizes, and it makes you feel safe. You glance at a shaker shelf where flasks sit with their long necks topped by soft stoppers, the liquid swirling in a regular, rhythmic circle. The glass catches the light with a relaxed shimmer. The swirl continues, reliable and serene. It's like watching laundry in a slow cycle, the same movement repeated in a way that never demands attention. You could watch it for a long time, letting your thoughts float over it the way a leaf floats on a current. The gentle rhythm continues. Now imagine you are at a small bench, across the plate with a sterile spreader that glides over the surface like a skating blade over fresh ice. The plate is set aside to rest in warmth. Later, when you return, you count with a clicker in your palm. Each soft press a small tally of time lived. 1, 2, 3, 4. Not numbers as a demand, but more like counting the strokes of a cat's fur. The numbers add up to a concentration, an understanding. But right now, they are just soft taps in the quiet. Elsewhere, a sequencer hums at a pitch just above silence. You think of the information it reads like a library of letters arranged so patiently that reading them feels like listening to rain. Each letter is a drop, each drop part of an endless soothing pattern. The resulting sequence does not burrow into your mind with urgency. It rests there peacefully. Another entry in the record, another line in the notebook that says, gently, this is what was here, at this time, in this place. You refill a wash bottle, the clear stream of water running along the inside wall and falling without splash. You return it to its place, angled nozzle aligned with the others, beads of water still clinging near the tip like tiny pearls. The gentle rhythm continues. You take a clean cloth and fold it into a neat square. You align plates in a rack so they sit evenly. You straighten a small stack of printouts, the paper whispering as you slide the edges into alignment. These motions are not distractions. They are parts of the same care that guides the pipette and the loop. Care, repeated, becomes its own lullaby. In your mind's eye, you place a gentle thread between this quiet room and the open world. You picture soil crumbling between fingers, each crumb packed with internal passageways and pockets where communities settle together. You see the underside of a leaf, soft and pale, with a hidden neighborhood of tiny residents who live as tenants in a calm arrangement. You imagine a river rock, smooth and cool to the touch, its surface a home to film thin layers. Each scene is peaceful and normal. The world has room for these lives. It always has. Now, as your focus returns to the bench, the second half of your day begins to fold inward, like pages closing on a story that has been read gently and deliberately. Still awake, comment the secret word, nebula. You move to the gel box, a small tray waiting for an even bed of gel. You pour the cool liquid that will set into a clear, firm cushion. Later, when you place samples into wells, pipette tips slide softly into the small rectangular openings, and the liquid settles in like calm dew, pooling in hollow leaves. The gel rests beneath a quiet lid. The buffer, transparent and faintly bluish, fills the chamber with a soft gleam. When the current starts, bubbles rise in slow, tiny beads from the electrodes, a dreamy stream that almost looks like a miniature underwater garden. You imagine the pieces of DNA gliding through the gel like gentle, invisible swimmers. Time passes, and bands emerge in a muted glow, lines of light forming like constellations on a clear night. You watch without urgency, letting your mind drift. When you finish, you clean each part in a thorough sequence. The comb is rinsed and dried, the tray is wiped, the platform is restored to order, a cap unscrews and screws back with a tiny squeak that feels like a soft exhale. You're not rushing to the next step, you are simply making room for peace. You turn to a small refrigerator to retrieve reagents, the door opening with a hush and closing with a gentle seal. Bottles line the shelves like patient sentries. The labels face out in tidy rows. You lift one, read its name quietly to yourself, and return it with care. You feel the cool air brush your hands, and then it is gone. Light shifts in the room as the day leans toward evening. The overhead lamps keep their soft, even tone, but you sense the outside world dimming. It feels like the lab is slowly closing a curtain, inviting you to wrap things up calmly. There is no pressure, only closure. A stack of plates that finished their day's growth is ready for storage. You turn them upside down to prevent drips from disrupting the surface. Their lids snug. They rest in a bin, labelled and dated, like a file drawer of small, serene planets. Before you end, you perform one more check. You look in on a slow ferment, a jar with a gentle balloon of gas indicating a quiet transformation within. You smile without thinking. There's something soothing in an invisible process made visible by a simple, gentle sign. The gentle rhythm continues. You tidy the laminar hood, letting the clean wipe sweep under your palm with smooth strokes. You dispose of used tips, their faint colours gathered in a small container, then closed and set aside. Around your wrist, the elastic of your glove feels easy. You peel it away and find the air cool on your skin. You wash your hands with warm water that flows steadily, the soap creating a light lather that slips away as easily as a soft fog lifting from a field in early morning. You dry them with a towel that rustles like a page turning. You breathe out. You are ready to let the day rest. As you sit with your notebook, you draw a final line under your last entry. You add a calm note, incubating overnight. Check in the morning. You close the cover with a subtle thump that feels deeply satisfying, the way it feels to make a bed and smooth the blankets. The lab continues its quiet work without you. The incubator hum remains. The shaker's gentle wave repeats and repeats and repeats. The gentle rhythm continues. In your thoughts, you walk through the day again, but in softer steps. You poured plates, and the agar set. You streaked, and the colonies began. You counted softly, and the numbers made sense without command. You used a pipette like a metronome of care, and the plunger pressed smooth under your thumb. You listened to equipment that never raises its voice, and you responded in kind. Even the stains and the gels felt like gentle watercolor and soft glass. The hour-by-hour tasks blurred into a single continuing tone of calm attention. You think of how this quiet craft shapes the idea of patience, not as a burden, but as a duvet of time that warms you from shoulder to foot. You are never at the mercy of a loud clock here. Instead, you participate in a slow unfolding, like a patient story told by a soft-spoken friend who never rushes a sentence. The results come as they always do, gradually, like dawn. You find that even your breathing has learned this, taking its cue from the incubator's subtle hum. Your body recognizes it and becomes still. Imagine now that it is late and the lab is dim. One light remains on at the bench, its circle of illumination gathering a few gentle shadows. The room's edges grow softer, the corners more muted. You stand by the incubator and watch your reflection faintly in the glass, a calm figure held in place by the warmth behind the door. The stacks of plates are steady and sure. The loops and pipettes are clean and at rest, ready for another day. The floor is swept, the trash is tied neatly. Everything speaks in a low voice. Tomorrow, the gentle rhythm continues. You think briefly of all the people who will take quiet comfort from this work in ways they may never notice. A loaf with a good crumb, a garden with healthy soil, a ferment that gently sings in the kitchen, a classroom where a student finds, in a small colony, a private, beautiful sense of possibility. These thoughts are not grand or heavy. They are small and reassuring, like the thought of a porch light left on at night. Before you go, you step again to the microscope, just for one last look. You place the slide with the ease of a familiar routine. You turn the focus and watch the edges sharpen until the tiny shapes become crisp once more, like seeing snowfall through a window in a quiet house. You feel, for a moment, how soothing this scale can be. It makes room in the mind. It creates a breadth of stillness that lets thoughts drift and settle wherever they like. There is goodness in that drift. There is rest. You click off the lamp and return the slide to its case. You set the case in the drawer with the slides that will wait until morning, lined up like peaceful pages in a book. You close the drawer gently. The bench is clear now, and the room is as it should be, clean, steady, complete for today. You walk toward the door, hearing the soft, familiar sigh of your shoes on the floor, the distant, steady breath of the incubator, the whisper of cool air from the vents. You pause with your hand on the handle. The day ends not with a period, but with a long, friendly ellipsis, the unhurried promise of continuation. You step out, letting the door find its corner quietly. As you move down the hallway, you realize that the patience learned in that room comes with you. The world outside will have its pace, as it always does, but you carry the slower heartbeat of the lab within. Your thoughts will remain orderly without forcing themselves. Your breath will keep its new rhythm. The gentle memory of pearly colonies and smooth agar and steady hum will stay with you like a soft melody repeating in the background. When you lie down later, you might recall the hush inside the laminar hood, the slight press of a pipette's plunger, the quiet clarity of a glass cuvette, the untroubled growth of a pale colony circle under gentle light. These images can settle your mind like snow in a globe, drifting in layers until everything inside is still. If you need comfort, imagine once more the room in its quiet brightness, the bench top reflecting a patient glow, the incubator door closing with a soft seal, the the notebook resting with its cover closed and its spine aligned to the edge of the table. Imagine yourself there, breathing evenly, supported by routines that ask for care and give calm in return. The gentle rhythm continues. Thank you for spending these patient hours with me in the soft company of cultures and light. If this calm journey helped you drift toward rest, a quiet like and a gentle subscribe can help us release more of these soothing episodes as often as you need